Hello everyone, welcome to today's class. Today we are going to see class 9 science chapter 2 cell, the building block of life. So let's start with the learning objectives. So this chapter helps us to understand what a cell is and why it is important, and also types of organisms based on the number of cells, and how body organization occurs in living beings, and also how scientists study tiny cells. Actually, this understanding cells is very essential because they form the foundation of all living organisms. Every structure and function in the body begins at the cellular level. Without studying cells, we cannot understand how tissues, organs, and organ systems work together. Cells carry out vital processes. What are the vital processes? Already we saw in our previous lesson, right children? Like respiration, nutrition, excretion and reproduction. By learning about cells, we gain insight into how life functions and how organisms survive. For example, if we take like doctors study blood cells to diagnose diseases like anemia or infections. Our scientists, they study about the nerve cells in order to understand the brain function and also communication in the body. Let's start with the topic today. Before entering into the cell, you have to understand from where the life originated. So origin of life, where it happened children, it started in the water. So it is widely accepted among scientists that life originated in water. Early earth conditions, it was around 3.5 billion years ago, were very different from today. It was completely different. Some researchers suggest that life began in small water bodies like hot springs rather than large oceans, because such environments, they had a fluctuating conditions that supported the chemical evolution. For example, we can say a very good example is hot springs in Puga Valley, where temperature remain extremely high even in a cold climate. These conditions, they are very similar to early earth. Yeah, and the starting how the earth was. So organisms found here, they were mostly thermophiles. What is this thermophiles children? They can live in extreme heat temperature. Here thermophiles, thermo means heat, right? So they are heat loving bacteria. They are unicellular. So the scientists from the Birbal Sani Institute of Paleosciences, they discovered that calcium carbonate forms rapidly in these hot springs. So they were able to find the deposits of this calcium carbonate, and what it did, it protected the early organic molecules from harmful radiations and also helped in forming the first cell membrane. So, for example, like thermophilic bacteria surviving in extreme heat was found, and are also mineral deposits protecting early life forms was found. Let's move on with the topic. Yeah, let's start with the topic today, the cell. What is a cell, children? See, the cell is the basic unit of life. All living organisms are made up of cell. We can say a cell is the smallest unit, yes, which is capable of performing all life processes, yes, of course. And what is it? It is making it the structural and functional unit of life, because it is performing all the life processes. So it is having a proper structure and is also performing so many functions. So it is a structural and functional unit of life. So here two ways organisms are built. One is a unicellular form, another one is a multicellular organisms. What is this unicellular organism? Say they are made up of, consist of only one cell. They perform all life activities. Yes, for example, like bacteria, yeast, amoeba, and then these are even the thermophiles, yeah, in Poga Valley hot spring, they are also bacterial. So they are unicellular forms. Multicellular organisms. See, these organisms consist of many cells working together in a coordinated way. For examples like plants, fish, birds, humans, yes. So they have a like you can able to see here cells, tissues, organ system, yes, organ, that a level of organization you can able to see here. I will teach you what are those things in the next slide. So examples are like a respiratory system, nose, trachea, lungs, all these are some examples. So all these things, no, children, did you know that scientists believe life originated in water, possibly in hot springs, those in Puga Valley, Ladakh, yeah, everything, just go through all these things. These are very important points. Next, we are going to see uh, levels of organization in living organisms. See, when we talk about the multicellular organisms, cells are organized into different levels. First level, the basic unit is the cell. Yes. So the cell, they group to form tissues. They are having similar cells. Yes, tissues are nothing but group of similar cells. And again, organ is formed. So different tissues working together, they form the organ. And then what is this organ system? Organ systems are nothing but group of organs. And at last comes the organism, which is a complete living organism. So real life example also I have given here. So what happens in the respiratory system? Nasal pores, through the nasal pores, yes, where the air is going to the nasal cavity, from there it is going to the trachea, from there it is going to the lungs. So all these work together, help us in breathing process. Yeah? So the life remains the fundamental unit of a structure and function in all living organisms. Yes. So these are very important, children. Yeah, you'll get some questions from this part also. Let's start with the next slide. How to study cells. See, cells are the basic units of life, but most of them are extremely small and cannot be seen with the naked eye. So this raises an important question, like how do scientists study such tiny structures? To answer this, we must first understand the limitations of the human eye and also the tools used to overcome them. So what is this limit of resolution, children? See, limit of resolution is nothing but the ability of the human eye to distinguish between two very close objects. Yes. So this ability we called as the limit of resolution. How to define it? The limit of resolution is the minimum distance. What is it? Is the minimum distance between two objects at which they can still be seen as separate and distinct. So I have given here for the human eye, this limit is approximately how much? 0.1 millimeter when viewed at the near point of about 25 centimeter. So what this means? This means that if two objects are placed closer than 0.1 millimeter, they will not appear as two separate objects. Instead, they merge and appear as a single point. For example, we can say like two tiny dots, yeah, drawn very close together will look like one dot. And then we can say like a fine structures like cells also cannot be distinguished. So this limitation explains why we cannot directly observe microscopic structures. Why can't we see cells, children? See, most cells are smaller than 0.1 millimeter. So they fall below the resolving power of the human eye and they remain invisible without assistance. Let's see the size comparison. For example, if you take onion cell, it will be around 100 to 300 micrometers. When you take a bacteria, it will be 1 to 10 micrometers. When you take viruses, it is even smaller. So it will be measured in terms of nanometer scale. So these sizes are far below 0.1 millimeter and the cells cannot be seen without magnification. So in order to study these cells, we need special instruments like microscope that can magnify and also resolve such tiny structures. How do we see cells, children? See, scientists use microscope to observe cells and other microscopic objects. How does it work? See, microscope use convex lenses to magnify objects. So these lenses, what they'll do, they bend the light rays to produce an enlarged image. We have two lenses there. One is the objective lens, right? And another one is the eyepiece lens. So what this objective lens does? See, objective lens is placed close to the object and produces the first magnified image. And this eyepiece lens, it further magnifies the image of the observer. So together both these lenses, they make tiny objects appear much larger and clearer. Okay. And then next we are going to see we are going to understand the size scale here. Can you able to see, yeah, the diagram here? So this diagram is the size of objects and visibility from unaided eye to microscope. So what it represents, it helps us to understand or compare objects of different sizes, from large visible objects to extremely tiny ones or tiny particles. Can you able to see the large visible objects here, children? So here the rockets, yeah, these objects are large. It can be seen with your eyes, yes. Rocket, you can see trees, you can see human beings, or these birds, and also chicken egg. Microscopic objects, they need microscope. So these are too small to be seen with the naked eye, but can be observed using a microscope. What are these? See, the examples are amoeba, most plants and animal cells, nucleus, most bacteria, mitochondria. Yes, all these are some examples. And then ultra microscopic objects, they need powerful microscopes. So these are extremely small and require advanced microscope. Like examples are like viruses, ribosomes, this protein, lipid molecules, and also even small molecules and atoms are some examples. So this slide helps us to realize that the human eye has limitations and many important structures of life also exist beyond what we can see directly, right? So microscope, they act as a powerful tools that extend our vision and also allow us to explore the microscopic world. Yeah, let's start with the next one, the light microscope. So before getting into this light microscope, let us study who discovered this cell. See, cells were first discovered by Robert Hooke in 1665. He observed a thin slice of cork under a microscope and he saw small box-like structures which he named cells. Okay, let's start with the microscope. So this is the diagram of a light microscope. Yes, can you able to see the diagram, children? And parts of the light microscope. You can able to see the main parts. See, this is the eyepiece. Yes, this is the eyepiece. Body tube, you can able to see these objective lenses and stage. And this is the mirror. And this is a coarse adjustment knob, fine adjustment knob, handle, and the base. Okay, all these things are very important. See, children, this eyepiece and the objective lens, we can change the resolution of the objective lens. For example, you'll be knowing your mobile phones. You used to buy different mobile phones. Like why you are buying? Because of its camera clarity, or it is having more megapixel. Like that, you used to buy, right? Like this here also, we have this lenses, magnifying lenses. So the total magnification can be controlled with the help of these lenses. Yes. So how to do this? See, for doing this magnification, we have a magnification formula. That is, the total magnification is equal to, total magnification here I have given is equal to the eyepiece into objective. So for example, if the eyepiece lens is 10x, yeah, and the objective is also 10x. So eyepiece multiplied by objective. So 10x into 10x is 100x. Yes. So we can change in order to get a better resolution. And we also have different types of microscopes also. Yeah, like we have a light microscope, yes, fluorescent microscopes. Yes, generally in the school we used to use a simple microscope and compound microscope. Yeah, in the research center and you can able to see the light microscope, fluorescent microscopes. Yeah, different types of microscopes are available. Yeah, let's start with the next one. So here we are going to see one activity. So estimating the size of a cell. So how do scientists measure something they can barely see? So here is a simple method using a microscope and a onion peel. What is the purpose of the activity, children? See, cells are too small to measure directly. So scientists use a microscope method to estimate their size indirectly. So this activity shows how to calculate the approximate size of a cell using the field of view. Yes. So let us see the step-by-step procedure. You get a ruler. Yeah. What is a ruler? Some used to say scale, right? Take a transparent ruler, yes, with millimeter markings and place it on the microscope stage. Why? This helps to measure the size of the visible area. Then what you should do? You have to measure the field of view. So focus the microscope on the ruler first. Then observe the diameter of the circular field. That is what you see through the eyepiece. For example, if you see 5 millimeter as the diameter means, next what you have to do, you have to convert to micrometer. So how to convert? See, 1 millimeter is equal to 1000 micrometer. So 5 millimeter will be equal to 5000 micrometer. Can you see here, children? Yes, all this. So 5 millimeter is equal to 5 into 1000, that is 5000 micrometer. Next, what you have to do, you have to mount onion peel. The fourth step is mount a onion peel. So remove the ruler, place an onion peel slide on the stage. Then you have to focus clearly. What you will see, onion cells are easy to observe under a microscope. That is why we are using it here. So count the cells. So count cells along diameter in one straight line. Yes. Then what you suppose you count 25 cells. Yeah. So we have to find the size. So estimate the size of one cell. We have to use one formula. What is that formula? See, let us take the field diameter. How much is the field diameter, children? Field diameter is 5000 micrometer, right? So how many cells we saw? In that number of cells we saw is 25. We have to find the estimated cell size. How to find? So cell size equal to, yeah, what is that formula given here? Diameter of visible field. Can you able to see? Diameter of visible field divided by number of cells along diameter. What is the diameter of visible field? That is 5000 micrometer. And what is the number of cells along diameter? That is 25. That is 5000 divided by 25 is equal to 200 micrometer. That is the size of a single onion cell. That is a one onion cell. Yes, like this we can measure the total visible area and we can divide it by the number of cells. So this will give the estimate of the size of one cell's, children. This method is very useful because it helps us to understand the microscopic measurements clearly. Yes, let's move on to the next slide. Here, apart from, I told you, na children, in school we'll be using compound microscope, simple microscopes. Yes, in when you go to some research institutions and yes, universities, you can able to find this electron microscope. See, this maintenance of electron microscope is not that easy. So, universities will be holding this electron microscope. So to see the structures even smaller than what light microscope see, so scientists mostly they opt for this electron microscope. So light microscope is electron microscope. Let us see some difference between it. What is the source of light? In the light microscope, it has some visible light. Electron microscope, beam. Electron beam is the light source. And scale of view. Here it is measured in terms of micrometer. Here it is measured in terms of nanometer. Very small, right? So here they use in school labs and yeah, everywhere, wherever possible. Mostly in school labs and some colleges. Here, electron microscope, it will use the fine cell details, children, actually. That is why they are using in research institutions or in universities. Yes, they opt for this electron microscope. So what is the three key features which has been improved over time? One is the resolution. Yes, like I told you before also, we used to buy new mobiles in order to get a higher resolution, or cameras, higher resolution, and also to get yes, that magnification, everything, right? So here also the same thing, resolution. That is the measure of clarity is very high. Contrast is brightness difference is very nice. And magnification, we can enlarge it to the atomic level we can able to see with the help of this electron microscope. Yes, let's move on to the next topic. Here I have given you some memory tips and also easy ways to remember. So Hooke plus Clark. Yes, who is that Hooke? Robert Hooke, right? Robert Hooke in the year 1665 he discovered the cells, right? So Hooke, where is in the Koch cells? What he found? Cells. So Robert Hooke saw Koch's boxer like spaces in 1665 and he called them as cells like rooms in a monastery, right? So Hooke peeked at Koch cells. And then resolution. How is the resolution? 0.1 millimeter. Hold this book at 25 centimeter from your eyes. So that is the near point. So anything closer than 0.1 millimeter. So what happens? The two dots become as one, like a thin pencil tip. And then magnification. So total, how you will magnification, how you can say is like eyepiece multiplied by objective lens. So like a eyepiece 10x and a objective lens 10x hi-fi, that is 100x. So multiply, yes, don't add children, multiply. Light microscope with electron. So light is measured in micrometers, that is only mu they have given, and electron in the form of nanometers, so small n. Yes. So light microscope works in micrometers and electron microscope works in nanometers, nm. So L is for larger units and E for even smaller ones. Yes, let's move on to the next one. So here given some NCERT questions from the textbooks, children. All these questions are already there in your textbooks. We'll have a separate video for these questions and answers also. You can go through, right? So we have come to the end of this topic. So what we understood from this lesson, see, actually, we spoke about the cell, so how the cell originated, and we saw cells are the fundamental unit of the life, and from the origin of the life in extreme environments like a Puga Valley to the discovery by Robert Hooke. So, and also what we saw, use of advanced microscopes is our understanding has greatly improved, right, children? So, since cells are too small to be seen by the naked eye, microscopes are essential tools that help us to explore the microscopic world. So we have come to the end part. We'll see you in the next class. Yes, please like and subscribe the video. Yes, so keep in touch. Watch the full video, children. Thank you.